So in this chapter, we're going to look at how we diagnose infectious diseases. Don't feel like you have to understand every technical step of every test that we talk about. Focus more on the advantages and disadvantages and example diseases that we diagnose using each test. There are a variety of tests that a clinical sample would go through when they go to the lab. In this form is sort of the old school method where we can see all of the possible tests that might be run. Of course, today everything's electronic, but the results of these tests can indicate microbial infections, and the type of samples that are sent just depends on the symptoms and what disease is suspected. Usually it depends on the test. There are some tests like the rapid strep test that can come back within minutes. They can get at least a preliminary result. They might actually send that sample off for culture as well, which might take longer. And of course, tuberculosis, remember that's the mycobacterium. It has a waxy cell wall, so it takes several weeks to culture tuberculosis bacteria. And of course, it depends on which infection is suspected, what kind of clinical samples might be taken. So the proper collection method for any clinical sample really depends on where the specimen is coming from. We want to avoid contaminating the sample, of course. And we want the sample to be a true reflection of the possible problem. So for example, a urine sample, what they do is called the clean catch, to initially wash away any bacteria that's in the urethra and the opening of the urethra that may not actually be the organism that is infecting the internal organs. And so most of the tests we're going to talk about in this chapter fall into one of three categories. When we say it's phenotypic, you can kind of think of those things that are observable, right? Those microscopic and macroscopic things that we can see under the microscope, things like their morphology, or even biochemical tests where we're just looking for color changes. We're going to have tests that are immunologic in nature, which means they're going to rely on how specific antibodies are against the antigens that they have affinity for. And with new advances in the Human Genome Project and the Microbiome Project that we've talked about, there are a lot of new methods that are using genotypic assays. And so they would look basically at the sequences of DNA and RNA of the microorganism to identify them. So many of the phenotypic methods are actually things we've already covered this semester. So for example, direct examination, putting a grammed stained slide under the microscope and determining morphology and arrangement. We've grown organisms in the lab using selective media, where you're looking for changes in the colony color or just whether they grew or didn't grow. Some of your unknown ID labs, we're using biochemical tests where you're looking for the evidence of an enzyme of some sort and just looking for a color change in the media. We did the Kirby-Bauer test, which is an example of susceptibility testing, where we're looking for antibiotic effectiveness. And then we talked briefly about things like phage typing, right? Using bacteriophages to actually detect viruses, or using cell culture to grow viruses because they can't grow without a host cell. So most biochemical tests use some sort of a color indicator, looking for the presence or absence of an enzyme. So for example, think fermentation. If a bacteria is presented with the right substrate and the enzyme is present, then it will produce an acid. And a lot of the pigments used in these color tests are pH sensitive. And so it will change color if the pH of the media changes. So of course, in the clinical setting, it's important to get a diagnosis as quick as possible. And so most of these biochemical tests today are done on different kinds of plates where they can do multiple biochemical tests all at the same time with one incubation period or sometimes no incubation periods, where they're looking for those color changes. And then they would compare that array of results against a control panel, and they would look to see if it changed or it didn't, and it would give you essentially the identification of the organism with the combination of all the test results on one array. And of course, we've used the dichotomous key as well. You have to have these results from the lab in order to follow the flow chart that helps us to identify the specimen in question. So again, this combines both phenotypic results of looking under the microscope as well as those biochemical tests. So it's usually a combination of different forms of tests that leads us to the proper answers. So in many cases, we can use the high affinity between antibody and antigen to then diagnose the infection. And this is really important for those organisms that we cannot culture in the lab. So we're going to take a look at each of these types. So we take advantage of the fact that the human immune system can produce very high affinity antibodies against the antigens that we get exposed to. So we can use a serum sample to go searching for the antibody as an indication that we've been exposed by using prepared antigens. Or we can go searching for antigens floating around in the serum by using prepared antibodies. So this is generally how we look for all the different kinds of Salmonella species. There are many kinds. There are dozens of species that can cause pneumonia or meningitis that fall into these two categories. And remember that many of these we can't tell the difference either by staining or other tests that we know how to do. But we do know that the human immune system can tell the difference between all these different serotypes. And so that's how they get the name serotype. Sometimes you'll hear they use the word strain as an indication of a different kind of bacteria that falls under the same group. We can identify fungal and amoebic infections. In some cases, HIV and syphilis tests will use this same idea of antibody-antigen affinity as well as what we do with measles and rubella. So we're going to see different varieties of tests that use this same idea. So agglutination tests look just like they do how we determine blood types. It's all based on the affinity between antibody and antigen. And if they find each other and bind, we call this cross-linking, and they will fall out of solution, and then a machine can read the color variants on these plates. And also a big advantage is that when we do a serial dilution, we can then determine the quantity of antibodies present, or what we call the titer, and that's usually defined as the lowest concentration of antibody that is detectable. And so this is a very fast test, but it has fairly low sensitivity compared to some of the other immunological tests that we're going to look at. But it is helpful to diagnose certain diseases like leptospirosis, congenital toxoplasmosis. You can diagnose this very early in a baby that has been born to a mother with chronic infection, so the treatment can start immediately. Brucellosis, which is a disease we vaccinate against in cattle, but it can be diagnosed using these tests, as well as parvo in dogs. So another type of immunological test uses pigments, and it's called immunochromatography. The most common one is what we call the lateral flow test, and this is where a little plastic cartridge has been impregnated with the antibodies that match the antigen in question. And then the sample is loaded, a reagent is applied, and the fluid will then draw through capillary action, will draw that sample across the antibody and the pigment. And so if the antigen is present, then an immune complex will form, which is just an antibody-antigen binding, and a visible reaction will happen when that pigment then is interacts with those immune complexes. So this is a common way. This might also look familiar. This is how we do home pregnancy tests. They're just using antibodies that match the hormones of pregnancy. This is also how we do the rapid strep test. So this is the one that you can get back the answer within 20, 30 minutes. They'll usually follow that up with a culture, but the rapid strep test is done just like this. They can also diagnose gonorrhea this way. There are a number of HIV tests that can be purchased and conducted at home. Some of the easiest ones are like this. But of course, these use a saliva sample typically, and they're not as accurate as a plasma sample. So we'll talk a little bit more about HIV tests here coming up. So the Western blot procedure has been around for a really long time. This is where you basically extract proteins from a sample and then you run it across a gel using electrical impulses, so it pulls apart and separates all the proteins. And then you transfer those to a membrane that then gets applied the antibody against the antigen that you're looking for. And that antibody is tagged with a color pigment, and then that pigment shows up. And so you can compare the patterns. And so each step is very simple, but there are so many of them that it introduces too many opportunities for error. So if this is being used for diagnostics, it's got to really be done carefully. And so it's usually used as a secondary test, and it's what we use to confirm if you have an ELISA test, which we're going to talk about here in just a moment. If you have an ELISA positive test for HIV, for example, they would definitely do a Western blot to confirm that diagnosis. Hep C and syphilis, those are also sort of backup tests. And so, of course, now, especially with the genetics tests that we're going to talk about, there are much more sensitive tests available. And so it just depends where you are, what kind of clinical lab is available, if they're going to still use this as the backup test. In fact, in 2014, the CDC was seeing so many false negatives if the person had just been exposed. So if they're earlier than 3 months post-exposure, then this test really gives way too many false negatives. And so that's when CDC said, no, we've got to go to a genetic test as the backup plan. The other disease that we use this for in combination with the ELISA is Lyme disease. This is a notoriously hard to diagnose disease because it can set up as a chronic infection and leave you with symptoms that are that may be years between when you have the acute initial infection and the later infection. And so one thing that is advantageous is because the ELISA is so much more sensitive. If you have a positive ELISA test but a negative Western blot, it may indicate that you are in fact infected, but you're in the asymptomatic phase. And so that can also help with how they treat the symptoms involved. Fluorescence is one way that we can tag antibodies with something that the microscopes have really high sensitivity to. And so if we use this, it's usually for those organisms that we have a hard time growing in culture. So syphilis is one of those that we've known about for a long time, but we've only recently learned how to grow it in culture. And so this has historically been the way that we diagnose syphilis. We have antibodies that are very specific to this spirochete, and it shows up real obvious on a fluorescent test. There are many strep species, but what we call the group A strep, or the gas group, Streptococcus pyogenes, is one of the most virulent strains, and so they are using this now to detect those. Mycoplasma, you may recall, that's the one bacterial group that does not have a cell wall, and so it makes it hard to do gram stains and get accurate results, and they're also notoriously hard to culture. And so if you want to detect or diagnose walking pneumonia, this is one of the tests that they will do as a more confirmatory test. And even measles is a virus that when it infects the cells, it creates these things called syncytia, where it basically causes neighboring cells to just merge together and make these big giant cells. And so those syncytia actually display antigens that are easily detected by antibodies, and then if they use fluorescent dyes, they can determine how many, or the presence of these syncytia as an indication of measles infection. So what is an ELISA? It's an enzyme-linked immunosorbent assay, which up until very recently has been the preferred method for diagnosing a number of infectious diseases, partly because it's very quick compared to culture, which takes several days to weeks. It's very simple, so most labs can do it right on site. It's highly sensitive because of the affinity between antigen and antibody is so tight. It's very unlikely that you misdiagnose if you're using the right antibody. It's also much safer than what they used to use, which was called an RIA, radioimmunoassay, which then, of course, produced a lot of radioactive waste. So even though they used very low-level radioactivity in the form of tritium, it still produced a lot of hazardous waste that then had to be dealt with. So it's much safer today. Most labs have gone to enzyme-linked rather than radioactivity. Some of the downsides is making those antibodies on the front end can be very costly. It can be very time-consuming. And very oftentimes you'll have antibodies that don't have very high cross-reactivity, which means it can bind to more than one serotype of a particular antigen. And so you may or may not be able to distinguish between different strains of organisms if those antigens don't change very much from one strain to the other. The other problem is once you have the antibodies, those proteins are very sensitive to temperature shifts, and so they have to be refrigerated. So this is not something we can do remotely in third world countries, out in a mobile situation where you're on site at maybe a refugee camp or someplace where you're trying to diagnose certain diseases. That's not very practical. And there's a couple of different types. There's actually multiple types. We're going to talk about the two major types. So the indirect ELISA uses known antigens to go looking for the antibodies present in the serum sample of a patient. And so the serum sample would be loaded into this microtiter plate. If the antigen and antibody find each other, they'll bind. Then it's incubated with an enzyme that binds to the stem portion of antibodies. Then that enzyme will convert a reagent into a visible pigment, and that's how they can detect the presence of things like HIV. So this is the test we mentioned a minute ago. If this comes out positive, they would follow it up with a Western blot to confirm. But HIV is notoriously hard to detect early on in the infection, and so we don't have any tests that can detect it immediately upon transmission. The earliest we can find it and confirm it is using a nuclear test, a genetic test. But that is even at about 10 days that it's effective. Unfortunately, this test is also very expensive, and so they usually reserve it for high-risk exposure situations. So if you have a needle stick with a known HIV-positive individual, they might do this test initially. But usually it takes several weeks before you have detectable levels of antigens or antibodies. And so one of the tests that they'll do is called the P24 antigen test, and this is looking for this antigen that's on the surface of the virus itself. The other test is the one we've just talked about here, the indirect ELISA, and that's an antibody test, and that's basically looking for what they call seroconversion. And this word basically means that your immune system has now started producing detectable levels of antibodies. And so everybody's body produces antibodies at different rates. And so you can see here why the time course is so important. So people that order home tests, online, it's unfortunate if that's the only test they do, and if they do it too early, they're going to have a false negative because those tests are looking for the presence of antibodies. And look at the time course here, right? So it takes at least 2 weeks before your body can start making antibodies. And it's only about after 4 weeks you're going to have detectable antibodies. And then, of course, as the virus finds host cells, and then the immune system sort of settles down after that initial infection, that antibody titer will also go away. And so really, there's a window of time where things are most detectable. And so obviously if any of those home tests come out positive, they should be followed up with a clinical test as well. The other kind of ELISA is called the direct antibody ELISA, or sometimes they call it the sandwich test. And this one is much more specific because it actually uses 2 sets of antibodies, but it's also much more time consuming. There are many more steps. And so basically, just like the name suggests, the antigen, so this one is actually using known antibodies to go searching for the antigen in the sample. And so the antibody is loaded to the well. The antigen, if it sticks, will then be bound with a secondary antibody that is then enzyme linked and will then convert the reagent into the color change like we saw in the other one. So this is how we diagnose things like hantavirus, the measles virus, Hep B, Zika virus. When they were having the outbreaks, they developed one of these tests for Zika. Dengue fever, toxoplasmosis can also be diagnosed this way. But this test is actually much more diverse. They're using it for certain kinds of cancers, especially intestinal colon cancer, and certain pregnancy tests. The more sensitive ones actually are this type of test as opposed to that lateral flow test that we talked about earlier. So in some cases, we can actually do the test in the human body and look for a reaction that indicates that exposure. And so either an antibody or an antigen is introduced. So for example, the skin test that we use to detect or surveil for tuberculosis. This is actually just a protein extracted from the surface of the bacteria that causes tuberculosis. And that antigen is then injected under the skin, and then we wait for 48 to 72 hours to see if there's a reaction. And so this takes time, because it's basically you're seeing the cytotoxic T cells are coming in and doing the tissue damage wherever that antigen has been introduced. And so it can at least tell us that you've either been exposed or you've been vaccinated. And so doing surveillance for tuberculosis is really tough, because some people's immune system keeps it in check, and so they're asymptomatic carriers. In other cases, people will test positive because they came from a country where they regularly do tuberculosis vaccination. So we'll talk about some of the other tests and what is preferred when we get to the respiratory system diseases. So most of the modern tests are genotypic in nature, where we're going and looking for the genetic sequences of the organisms as an indicator of their presence. And so we won't look at all of these in great detail, but I will mention that what they call real-time PCR, sometimes you'll see RT-PCR, that is actually currently how the coronavirus test is being done. And there were some problems with it originally when they first released it, but the CDC distributes kits to different healthcare facilities around the country to do the test, and they have improved those tests dramatically. So one genetics test that's become very important in situations where time is of the essence. So for example, a high-risk patient for sepsis, like a burn patient, if they test positive for gram-positive cocci in the blood, you don't know if it's the normal Staphylococcus or epidermidis, which is a normal resident of the skin, or the highly virulent Staphylococcus aureus. And if sepsis is caused by Staph aureus, it has a very high mortality rate. And so one big advantage of the FISH test is how quick it is, right? Within 5 hours, you can get answers, as opposed to several days if you had to culture that bacteria. And it's also highly sensitive. In some cases, it can be 100% accurate in diagnosing which of these bacteria is causing the infection, especially in the Staph aureus range. But again, burn victims are highly susceptible to Pseudomonas infections. Candida, which of course is a normal yeast on the skin, if it becomes bloodborne, can become much more dangerous pathogen. And so just knowing which bacteria is causing the sepsis, and sometimes it is difficult to distinguish which organism is the problematic cause. This will make sure that when we prescribe antibiotics, that they are treating the proper organism. So in some cases, we have symptoms that are similar across many different diseases, especially when it comes to like the respiratory or GI diseases. And so because there's so many normal microorganisms that live in both of those areas of the body, most of the tests we have are not sensitive enough. And so this genetics test called a microarray can help parse out a particular pathogen that's present in either sputum or stool samples. Unfortunately, these tests are still very expensive, but they have been useful in cases where you have antibiotic-resistant strains that have a unique genetic sequence. So things like MRSA or multi-drug-resistant tuberculosis, these have been helpful. So in some circumstances, looking at the entire genetic sequence is going to be necessary to detect the cause of an infection. And this will be really important if we have outbreaks of unknown origin, or if we have new strains of drug-resistant organisms form. And of course, the technology's gotten better and better, and the cost has gotten lower and lower. And so it's not going to be uncommon that we do entire genome sequencing. And so usually what happens is they take the white blood cell, the DNA of a patient out of the white blood cell, and then they can match it up and basically extract everything that's human, and everything that's left should be other microorganisms. And then they can compare those to genetic databases that already exist. And so these should be quicker diagnoses. There's some advantage of having a genetic database that represents a local strain of an organism as opposed to what you see elsewhere. And so this is going to just expand our ability to diagnose specific diseases. The downsides, of course, is if there's a strain that we don't have in those databases, then it may not be recognized as a pathogen. It might be assumed to be a normal microflora. And then, of course, we know some viruses can insert themselves in our DNA. And so they're sometimes referred to as stealth viruses or latent infections. So things like human papillomavirus can hide out in our DNA in places that might appear as a normal gene in those strands. And so this is going to become more and more important as we try to detect diseases that are either fastidious. If you recall, that means that they require very special growth factors to be cultured, or species like tuberculosis, especially multi-drug resistant tuberculosis, that takes weeks to culture. In many cases, we're now finding that diseases involve actually more than one pathogen. So if we're using our traditional tests to define the pathogen, and we're not using something like genetic sequencing, where we can see multiple pathogens at once that may have required multiple kinds of tests. And so they sort of refer to this as an unbiased approach, meaning we're going to look for everything that's present, regardless of what kind of test we historically have used to diagnose that organism. So another way that we use gel electrophoresis is, instead of the Western blot where we separated out proteins, we're going to separate out DNA. And so what they call DNA fingerprinting is basically where they use enzymes that know how to locate certain genes and chop them up. And so then you basically put it through a gel where you have sort of an electrical field that pulses, and so it sort of drags and pulls the molecules apart. So they use this to determine which bacteria is responsible for some foodborne outbreaks. So it's very quick and is specific enough to find those species. In some cases, they're using it to detect bacteria that are causing nosocomial infections like Pseudomonas. There's a number of cases that were spread by endoscopes that were not properly disinfected in between patients. And so this is a newer technology. It's very quick and useful in those circumstances. So one of the newest technologies is essentially a microchip type of genetics test, where the chip is printed with a conductive ink that contains the genetic sequences of the pathogens that you're looking for. And then they have basically a chip reader that detects whether the pathogen sequence was found in the sample. So these are incredibly compact. They originally were called the shoebox lab. And the first ones they actually called the MR box because they were searching for measles and rubella. And so the manufacturers of those initial ones, they cost them about $50 a chip. Today, those same tests are only about $6 a piece, and the manufacturer predicts that they'll be down to pennies for each one by the time the technology improves any further. And so this gives a really quick test. You can get answers within 35 minutes in most cases, and so they're really helpful in places where you need a mobile diagnostic lab. So, for example, refugee camps where you have really crowded situations where you don't have any power, but you have an outbreak of some sort. And so diagnosing those is very important. So these are going to be really important as we move forward. But easy to use. They only take a tiny sample. For example, one single drop of blood can be used to detect a number of different diseases. There are very few supplies involved, very little training involved, and they don't require refrigeration, which is really important when you're going out to remote parts of the world to surveil for different diseases. So one of the oldest technologies we've used in science is mass spectrometry, which basically detects the mass to charge ratio of proteins. And so this is one of the quickest types of diagnostic tests we have. Within seconds, you have an indication of whether that antigen or the protein on that particular pathogen is present or not. So it's really important in places where, or in pathogens that we cannot culture. So things like SARS viruses, of course, require a host cell. And so SARS virus, they're probably developing one for the coronavirus now. It's very similar to the SARS virus. Ebola outbreaks, they've used this. Different strains of influenza can be detected this way. And some antibiotic-resistant strains of bacteria can be detected this way. The Acinetobacter species are really important in nosocomial infections as well. So those are notoriously hard to grow in culture, and so it makes it hard to control. They've also used this test for other diseases, not even just infectious diseases. So most labs have this technology because it's been around a long time, and so it's still being used to detect and diagnose infectious diseases. So other forms of technology can also be used. So imaging can keep us from having to do invasive procedures and biopsies. So for example, x-rays are always used to confirm tuberculosis. Even if the skin test came back positive, you want to confirm that with tubercles in the lungs. Other types of imaging we can use to look at deep tissues, for example, like an implant of some sort. If a hip implant becomes infected, that's a hard place to go in and take a biopsy. So this can help diagnose where those infections are. One company has actually even developed a system where the camera on your smartphone can actually be used to detect the fluorescence of a DNA reaction on a microchip like we just saw. And so these types of tests are really quick. So, within 30 minutes you can have that diagnosis. It's going to be really important again for places that are remote. So if you need mobile diagnosis, what they sometimes call point of care, especially if it's something like HIV infection, they're finding that if you have a diagnostic test that the patient then leaves and then needs to come back to get their results, very, very oftentimes they don't come back. And so if you can give them that diagnosis on site at the moment, then your treatment can be more effective. And in some cases, they need point of care because they are in a place where the disease is spreading, and they need to determine immediately whether a person needs to be treated or needs to be quarantined. And so things like the Zika, or in South America, we have a lot of dengue outbreaks that people, if they're quarantined, can actually keep it in check. So thanks to the Human Genome Project, we now know that there are certain genes that get expressed when a bacterial infection is underway. And so obviously if those genes are active, then antibiotics might be warranted, whereas if they're not, it's likely to be a viral infection. And so this is going to help a lot in our fight against antibiotic resistance by minimizing inappropriate use of antibiotics. Now, there are some other tests that they're looking into to look for genes that represent antiviral antibodies, because those, again, antibodies are proteins, and there's a gene that codes for that. So that's something that's underway.